Hey everyone, welcome to my channel, Anarchy for Freedom, in India's home for conspiracy research and free thinking. Today we're joined by molecular biologist Gabor Erdosi. I've been through his Twitter, I've actually heard a lot of his podcasts online, and he's a great wealth of knowledge on a lot of the subjects that we're going to be discussing today. So thank you so much for joining me, Gabor. It's a pleasure to have you here. How are you doing, man?
It's great. Thanks for inviting me. And
Maybe,
just go ahead and shoot any questions, and I will try my best to answer.
Sure. Maybe before we do that, if you could just introduce yourself to the audience, because we largely have an Indian audience and many people might not be familiar with you.
Sure. I have a master's in molecular biology. I also have an MBA. I usually don't tell it to people because I don't really do that kind of business anymore. I started my career as a kind of a medical sales representative. And among other things, we were selling, for example, personal protection equipment to operating rooms. Among that, masks, for example. And I participated countless education sessions given to surgeons and operating room staff how to wear masks, what masks are good for, and these kind of things. I was then a surgical product specialist in endoscopic surgery mainly. Then I moved on and worked as a technical account manager in industrial enzymes, and I became a technical support specialist also in industrial microbiology. And then I worked a couple of years in the starch processing industries as a sales support specialist, providing technical support for the salespeople, and also worked a couple of years as a product development manager. And for the last couple of years, I've been working as a micro entrepreneur, giving mostly R&D advices, recommendations as an R&D consultant to mainly small businesses, startups, and also some lifestyle type of consulting. So that's my background now. More than 20 years, almost 25 years of professional experience in various fields. So my hobby, kind of hobby, I would say it's an obsession more than a hobby. I've been reading into the science of metabolic diseases and the profiling of different metabolic markers, for example, insulin, and what you can get out of measuring glucose, different lipid markers, and these kind of things, and how the connection between lifestyle, for example, diet, and different diseases, and how to stay healthy on best practices, looking into the simplest kind of interventions. So how to avoid extremes. So the kind of the Pareto's principle to invest the least and get out the most with these interventions. So I have a lot of friends in India, for example, those who deal with diabetic people, type 2 diabetics mainly. And I think it's especially important for them to find a intervention which is easy to follow. It's not very restrictive, and it doesn't require very expensive foods, for example, or very expensive medication or supplements. So yeah, we have been discussing this with a lot of friends in India recently.
That's awesome. That's quite a bio, and it was really interesting for me because I was going through a lot of your videos as well, and many areas that I have like a passion in, and like have really dug deep into some of the things you share. Like I've really been digging up a lot of the science behind natural immunity, as well as PCR testing and all the controversies and debates surrounding that since the last year, as well as I'm deeply involved in functional medicine as well. So, both those things are very, very relevant to me. And probably we'll take up the aspect of diet and diseases later on. But I'd like to start with going through some of the science, mainly particularly surrounding natural immunity, as well as the testing, because I know that you've done a lot of work in those areas. And then probably, since you have a background in masks as well, and teaching people how to wear masks, we could probably touch on your views on that, because I see you were going back and forth on that issue with Kevin McKernan on Twitter. So maybe your views would be helpful. And then, of course, the subject of diet is very, very relevant to me, because I myself have been involved in the keto carnivore community, and I've been a vegan before that, and now I'm kind of more into biochemical individuality and advanced like testing to figure out what's really going on. So maybe we could have an interesting conversation out there as well. But why don't you just start with your views on the testing and like what you've learned so far in the last 1.5 years that this pandemic has gone on?
Yeah, partly because my small business partners experienced some issues when the pandemic started, or after that. I had a more than expected time to spend on reading into the virology and microbiology and the pandemic management part of this subject. So I literally read hundreds of studies, and I usually get my information from PubMed and Google Scholar and these types. So I read studies. I don't really read blog posts and mainstream media articles and this kind of information. I prefer to read basic science and draw my own conclusions based on the science what I find. So I read hundreds of studies, and fortunately enough, I had there was some overlap with my prior activities, because more recently I've been digging into mucosal immunity, mainly in the gut. So not the respiratory mucosa, but these are related on many levels. They are connected directly, but also there are many similarities how the immune system around this mucosal surface is arranged in the gut and in the respiratory system. So I had some clues, and then expanded my understanding with more specific readings into the respiratory mucosa. And I think it was very interesting at the beginning. First, of course, I also got the basic information from mainstream media that there was an outbreak in China, and there is this new coronavirus spreading. And then news came from Italy, and crazy footage of hospitals full with people, and even on the corridors there were people seen. And I was a little bit scared that it's spreading fast, and it's very deadly. And then gradually, after the first shock in early March, I started reading into the actual reports in the scientific literature, and then I started recognizing that somehow the data and the mainstream media reports there was a big discrepancy between these tools, basically. So I was a bit surprised, and based on the data, I already suggested in March last year that the kids should be sent to some kind of a camps or these what you have in summer vacations, and in hotels or wherever, where it can be done in a controlled environment. And they should just interact with some people who are already infected, and then get infected as soon as possible, so that they can provide protection for more vulnerable people. This was an idea already back in March last year. I got quite some ridiculing for this idea, but actually some 15, 16 months later, I think that this was still the ideal strategy back in March 2020, because there was no vaccine, there was no medication, effective medication in sight back then, and a vaccine had been something like at least 1 year ahead back then. So it was clear that we had to go at least 2 seasons without an effective preventive measure like a vaccine, and with the best medication we had. And then the data from these cruiser ships and Italy and China, what you could believe at least, showed that the disease is extremely kind of one-sided. So it's unevenly distributed between the elderly and the young people. And back then, it was not fully clear that it is based on so-called comorbidities, so pre-existing diseases. But it was clear that there was a huge age skew in this distribution. So young people already back then looked kind of non-vulnerable. So why don't just they acquire natural immunity with minimal risk, or even in a controlled manner? So when it turned out that obesity and other metabolic diseases were also risk factors, those kids could have been excluded, of course, from this immunization group. So that was my first idea back in March, and I still stand by that approach as being the optimal. And when reading into this, you could find supporting evidence that if there is a infectious disease which is unevenly distributed by age, sex, or pre-existing conditions, the mitigation measures should also take this into consideration. So that was the pandemic management plan, basically, it had been up to early 2020. And even some people say that Sweden, by following the 2019 pandemic plan management plans, they had the optimal approach to this. But I think that the optimal was kind of augmenting the old pandemic management plans with the uneven distribution of vulnerability. So that was the straightforward approach what I saw already back spring last year. And what actually happened was just the opposite. So I like to call this approach lawnmower approach, so applied to everybody the same way, while the susceptibility was not absolutely not the same. So I think it was highly unscientific from this perspective.
And isn't it surprising that like before the pandemic, the WHO in all their pandemic preparedness kind of documents, they never recommended any of the things that they're actually doing now. What do you think changed like after this thing began?
I'm not much into the political stuff of political background, but ultimately you start reading about different conspiracy theories that what happened and what the goals are with this crazy pandemic management, and I still don't know what is truth and what is not. But the funny thing is that more and more conspiracy theories come true over time. So what we believed a year ago to be a crazy conspiracy theory can easily come true this year. So it's,
I mean, that's the biggest case of that is the vaccine passports, right? Like I remember people were calling us conspiracy theorists
talking
about that last year, and now it's part of mainstream discourse, and it's being pushed.
Yeah. Also, the kind of denialism in the scientific community about, for example, natural immunity. So it has always been crystal clear that if you survive the infection, whether with mild or more severe symptoms, if you survive it, you get through it, you are immune. Otherwise, you should have died. If your immunity didn't work, you should have died. That's clear. Then you develop immunity. What kind of immunity? It's a matter of investigation. But studies have been coming out one after the other, showing that natural immunity was robust at 3 months, 6 months, 9 months, now 12 months and beyond. So now we are at 15 months, and the vast majority of people who built natural immunity following an infection are still immune, and they are not prone to reinfection. Actually, they are the least likely to spread the disease upon reinfection, and so on and so forth. So the denialism was crazy, and what I saw that most scientists were just silent. So they didn't contribute to the debate. What was clear that only scientists who were not really dependent on the financing background of governments, of big funding organizations, dared to openly tell what they thought. So, for example, retired immunologists and immunologists working in private sectors, not depending on the pharmaceutical industries or the testing industries. So it's a crazy thing that people now depend on the funding organizations, so that they don't dare to say what they think. It's just silencing, and those who told what they were thinking have been totally ridiculed. So they were.
This does line up with my own viewing into this over the last 1.5 years. I've myself, like you, have read many papers, especially on the testing matter, and I saw a big discrepancy between the media messaging and the narrative and what the scientists are actually writing in the papers. Because when people actually go read the scientific literature, they'll find many papers talking about the inaccuracies in the PCR and the pitfalls and the limitations, and the actual concept of infectivity and why PCR doesn't reflect that and things like that. So these kind of things are never really reflected in the media. So I always saw that, but I always saw that these scientists weren't vocal enough. Like, they would probably write papers or something, but when it comes to countering, when it comes to the practical application of their papers on reality, they were relatively silent in the public space. So this could be an interesting segue to kind of start talking about the science of natural immunity, because I mean, as we know, the WHO did try to fiddle with the definition of that a little bit. Like, they kind of removed the natural immunity part out of the herd immunity definition and then added it back later. So maybe, since your background is in molecular biology, could you give the viewers a basic primer on the immune system and respiratory viruses? Like, how does the immune system really kick in and work, both the innate and adaptive arms of the immune system? Maybe if you could just walk viewers through that, and then how that compares to vaccine-derived immunity and the benefits or pitfalls of both.
Yeah, I think it's actually very important to understand how the immune system at mucosal surfaces work. So when it's an interface between the outside and the inside, it's a very tricky situation where we have to maintain balance between proper immune responses. So overreaction is dangerous, because very high immune response can do harm to our own cells or tissues, and under reaction or a too weak reaction, of course, lets the virus spread all over the body. So maintaining this balance is crucial, and this is what this system has evolved for over basically millions of years. And there are some differences between how the system works in various animals and humans, but most mammals, for example, are rather good models. So when you see a study when they inoculate through the nose, for example, a mouse or hamster or a non-human primate. These are fairly good models, so it's not something you should dismiss immediately that okay, it's a mouse or it's a hamster model. No, it's fairly representative of, especially if you find the right balance of the viral load, how much virus to infect to simulate the level of disease typical in humans. For example, most humans develop mild symptoms with SARS-2 infection. So this is what the hamster model properly replicates. So what happens that there are different pathways how these viruses can infect a host, these respiratory viruses, but the most typical is that they are carried in so-called respiratory droplets. So when I'm talking or just breathing out air, some of the mucus, the thick liquid sticking to the surfaces inside our nose and throat, or even in the lung, leaves the respiratory system in small droplets. The size can differ. It could be just a few microns, or it can be bigger. Even what you can see sometimes that some small droplets leave people's mouths. So these droplets can carry a lot of viruses. And the interesting thing is that the smaller the droplet to a certain level, the higher the relative viral load in the droplet. So very fine droplets, so-called fine aerosols. You know, when you use a spray, for example, to odorize your toilet or whatever, these sprays let out fine aerosols. So that kind of aerosol, or when you smoke a electric cigarette, for example, and there is the smoke, that's also a fine aerosol. So these fine aerosols carry the highest number of viruses versus the droplet. So one tiny droplet can carry hundreds of viral particles. So you just inhale a couple of these droplets, and it can cause an infection. So this also leaves us with considerations about masks, for example, because the fine aerosols are not properly filtered by most masks. And if you have these very sophisticated respirators, most people don't know how to wear them. Then they leave gaps around, and then the only difference is that compared to open speaking or singing or breathing, the majority of the particles leaves not in front of you, but all around. So it's on the sides. It doesn't really reduce, at least not to a substantial amount. So yeah, and then you inhale these small particles. They stick to the layer which covers the internal of your mouth and nose and throat and lungs. It's called the mucus. It's a thick, sticky, barely fluid material, and it's kind of a primary trap for small particles, including dust and particles containing viruses, any kind of particles which should not directly have contact with your cells. So another tricky thing is that this mucus is not only sticky, but also contains so-called antimicrobial peptides. So things released by our cells into the mucus, which can neutralize different microbes. And on top of that, it also contains immunoglobulins. So these Y-shaped immune structures, which are specifically released to bind to certain microorganisms, bacteria or viruses, for example. and their specificity changes with your exposure to various microorganisms. And then these are so-called specific secretory. Secretory means that released outside. It's not circulating inside, but it's released on the outside into this mucus as a dimer, which means that two of these Y-shaped immunoglobulins are stick together, and they are stick together with a so-called secretory component. It's very important when from your it's called basal side of the cell, which looks internally, it looks inside internally in the body, on the blood side, basically. These immune globulins are released into the mucus. There is a band, kind of band attached, which contains specific sugar molecules, which make it more sticky to the sugar molecules on the outside of bacteria or viruses. And the specificity of these sugar molecules is very important, how they bind, because they can bind, so-called cross-link the virus to the mucus, for example. So they trap the virus by, it's kind of a specific glue. One side sticks to the virus and the other side to the mucus, and this mucus is continuously being elevated from your lungs, and you continuously swallow it unconsciously. So this is how it goes, that you continuously produce this mucus, this thick fluid, and it's being elevated. It's kind of an elevator in your respiratory system. There are so-called ciliated cells with tiny swipes upwards. So they carry this mucus up from your lungs to your throat, and you unconsciously swallow it and goes to the intestine. So this is how it works. And if this secretory immunoglobulin A, for example, can effectively cross-link the virus with the mucus, then you just get rid of it without the virus being able to infect you. If it reaches your cells still, so this mucus is too slow, it doesn't flow fast enough for different reasons, or your immunoglobulins are not effective, they are not very specific to this type of virus yet, then the viral particles can reach the cells lining the airways, and then they can effectively infect, so get admittance into the cells. And then when these viral particles are inside your cells, the cell starts producing kind of signaling molecules, alarm signals to other cells that there is some problem, something is taking over control. And if these signals effectively leave the cell, then immune cells recognize this. And one type of cells, the early cells, the early action immune system is called innate immune system. It's the most ancient type of immune system. There are specialized cells, and there are cells more or less specialized for every single function. So you have macrophages, for example, and neutrophil, different immune cells doing different kind of things. Macrophages can accelerate the immune response by producing inflammatory molecules further. They can so-called phagocytosis, so they can just engulf the particle and digest it. Or we have these neutrophils, they can leave out small traps, release some traps which bind to the microorganisms. And we have natural killer cells, which sense infected cells in the airway, for example, and then they arrive and kill the cell which is infected by a virus. So these can do it without any kind of prior learning. There are some kind of innate learning types called trained immunity, but let's leave that alone now. So this is the most basic immune system. We have a physical barrier, the mucus flowing. It contains molecules secreted by cells like antimicrobial peptides, which can directly kill or attach and inactivate to viruses or bacteria. And we have the cellular immunity among the cells covering the airways. And we have this immunoglobulin A, which is, funnily enough, part of the adaptive system, but kind of works together with the early innate immune response. So these are
would you have any additional insights on how the microbiome is involved in this? Because I've heard some microbiome researchers talk about how the microbiome is involved in actually messaging the immune system to come in when the microbiome detects pathogens.
Yeah. There is a microbiome covering the airways, and there is another microbiome more numerous in our gut. And both, interestingly enough, both are strongly connected to what happens in the airways. So there is a close interrelationship, a kind of a communication between the gut and the lung. It's called the gut-lung axis for this reason. And there is the local microbiome in the airways, which also strongly influence the outcome. So if you have a imbalance, for example, in the lung microbiota, then you can be much more prone to super infections. So when you get a viral infection, you are more prone to outgrowing of these opportunistic pathogens like streptococcus or staphylococcus and these kind of bacteria residing in the microbiome. So it's important to maintain a healthy microbiome, and if there is an imbalance, a pre-existing imbalance, that makes you more prone to bacterial infections when the immune system is busy with the viral infection. It's called superinfection because you get a secondary infection on top.
And I think that's really important to understand with the demonization of microbes in general that's happening today, that there are good microorganisms involved in our body in a very fundamental way that actually keep our systems healthy and keep us kind of safe from pathogenic or opportunistic microorganisms that could come and co-infect us, which is a narrative that's sorely missing today. I mean, even with the entire excessive sanitizing and just being scared of viruses and bacteria in general, that whole element is forgotten. But yeah, that's a very interesting explanation you gave of the innate immune system. So the innate immune system, just for the viewers to simplify it, is not specific, right? Like it's just the macrophages and neutrophils and natural killer cells are
different cells have specific functions, but the more or less the same cells can fight viruses and can fight different types of bacteria. So they are not specific in the meaning that we have the adaptive system, which develops much more specific responses and much more specific memory in the longer run.
So could you also go into the adaptive immune system and maybe talk about the interferon system?
I think that the role of the innate immune system is crucial in stopping respiratory viral infections early. So some people are known to be infected, being exposed to the virus, but they don't develop any kind of adaptive immune response. So some people have so effective innate immune responses that they can easily stop the virus before any adaptive response could develop. So it's very interesting, and I think that also the final outcome of COVID, the disease, the severity depends greatly on the initial innate immune response. If your innate immune response is robust and quick early into the infection, then you are almost certainly prone to a mild course of disease. So this is now clear, and just introduce the adaptive system, how that works. We have basically two types of adaptive cells, the T and the B cells. The role of the T cells is to organize the immune response further after the innate response started. So they direct the innate cells. They direct there are regulatory T cells, for example. They have maintained the balance so that there is no overreaction, but the intruder is efficiently cleared. So this balance depends on the balance of so-called effector, so working cells, and the regulator cells. The stronger the regulator, the milder the response, and the stronger the effector response, the stronger the cytokine release and the recruitment of effector cells. And the other type is the B cells. B cells are mostly responsible for releasing and specializing, adapting the antibodies, different kind of antibodies. There are earlier antibodies, bit less specific, like the IgA in systemically, and the IgM, and then the later more fine-tuned immunoglobulin is the IgG. This IgG is usually what's the longest lived, and this is what is measured when you get this antibody test in a lab or via a quick test. They look for the IgG developed against, for example, the spike protein. Then you can see after vaccination or after infection that you have immunity. Lack of this IgG is not indicative of total lack of immunity, of course. And so you can have a very effective innate immune system. You can have a very quick and effective T cell based immune system, and then you don't develop high amounts of antibodies.
In fact, I think in our diagnostic work at the clinic that I work at, we look at IgG antibodies for, I think, 28 or 29 different pathogens. Like Cyrex has a good panel that looks at all of these together. So out there, when we're kind of analyzing that, high IgG responses actually kind of signify a reinfection, or like something that's actually triggered back, and don't necessarily mean improved immunity or something, which is the context in which they're portraying it for this antibody test.
Yeah, so these T cells also can specialize to some extent. You have so-called killer T cells, the CD8 plus type, which work rather similarly to natural killer cells, the specialized innate immune cells in cell killing. So they sense and kill infected cells. And then we have the CD4 plus T cells, which can also kill, but they can organize the innate system, and they can also direct the B cells for a more specific antibody responses. And interestingly enough, what I've seen so far, the studies, people with deficiency in B cells, for example, who are there are some people with autoimmune diseases, or they have some inborn problem with B cell maturation or whatever. So people who lack B cells can also survive SARS-2 infection with mild symptoms. And there was a study which looked at, I think, macaques, so these monkeys, which were depleted of their T cells. And they were also able to tackle the disease with mild symptoms. I think that the only lacking of an innate immune system, responsive innate immune system, is which determines severe outcomes. So you most likely cannot lack both arms of the adaptive system, T cell and B cell based, but you can lack one of them without very severe outcomes. This is very interesting.
Would you have any additional insight on the role of B cells and T cells specifically for SARS-CoV-2? Like how are they really involved in neutralizing the virus and the different roles both of these play, and how they actually work to stop viral
Yeah,
infection?
it depends on what type of T cells you have as a kind of a naive person, because some of the T cells and in some of the people are fairly specific to SARS-2 even without being exposed, based on previous exposure to
Yeah.
human so-called cold coronaviruses. So they cause the common cold. There are four types of common cold coronaviruses which have which established themselves in the human populations hundreds or thousands years ago, and some of their proteins are not much different from the SARS-2 proteins. And the T cells can be fairly specific. It depends on individuals, it depends on previous exposure, how long ago it occurred, and what type of antibodies you have, or what type of T cell memory you have against these viruses. So it's not uniform. I believe that in some cases these can make worse actually the actual disease with SARS-2, because they are non-specific, but they are secreted in very high numbers, and they can screw the immune response. So they can make you prone to more severe disease. But in some cases, it just helps people to quickly and easily tackle the SARS-2 infection. So it's I don't think that it's uniform. Some people say that we had 90 something percent previous immunity. That's not what the numbers, not what the data say. So I don't think so. It's a more mixed reaction what we see in
So
real life.
I think it would be fair to say that we at least had some level of pre-developed immunity. Like the
Yes,
numbers would
yes.
probably be debatable.
But we, I think that in different populations there is also some level of more specific immunity to or against SARS-like coronaviruses, so a more closer relatives. But being exposed to, I don't know, bat crap or whatever, or other types of coronaviruses, there are some similar viruses even in Europe, but more in Africa and even more in Asia. So there was a study measuring this pre-existing immunity against SARS-like coronaviruses, and they found something like 1-2% in Europe, in Italy, I think, and they found 17% or something like that in Congo, in Africa. And I believe that it's even higher in Southeast Asia, for example, where these bats are living in higher numbers. So actually the exposure is more frequent and higher. So it can be, I don't know, 20 something, 30 something percent in Asia, where you have this even more specific pre-existing immunity. That could partly explain lower prevalence and lower susceptibility. Then there is, of course, a different age distribution, the demographics, and there was curious spikes in respiratory deaths in most Asia Pacific in 2018 and 2019, the seasons, which were considerably higher than the regular influenza seasons. So there might have been another less deadly coronavirus spreading before, and we don't really see population level antibody studies in these countries. So it's impossible to tell what the pre-existing level of immunity was.
Got it. So I think for a general layperson, this we're a little bit too deep in the weeds here, but just taking kind of a step back, this would the science of natural immunity would have two implications. One is, how do you think, like, what do you think would be the ideal way to actually measure immunity in someone? Because right now they're using antibody tests. So maybe if you could just touch on the cross reactivity, like what are your thoughts? Would the antibody test also react to other kinds of coronaviruses that already would be present in someone from before? So if you have any additional insights on the cross reactivity, and just looking at antibody tests to determine if someone's immune or not, with what we're discussing about innate responses and T cell responses, could there be a better way, like from a scientific perspective, that we could actually go about measuring immune responses in someone?
Yeah, I think it's difficult because, as we discussed earlier, some people, especially the most healthy, the healthiest types, can develop low antibodies at the beginning, and then it wanes over time. So if you would like to see 1 year later if whether these people developed long-lasting immunity, is very difficult. I mean, at least with a quick test or even with a quick lab test, it's not very easy. You could do a T cell test, especially if taking a sample from the throat or from somewhere a little bit more invasive, removing some little piece of tissue, and then do a T cell test. Otherwise, it's not that easy. So we have to rely on what we have, these lab-based antibody tests. There are
So what is the role of memory? I didn't touch of that, but the B cells also have memory. So is it scientifically accurate to say that if someone's antibody levels are declining, their immunity is somehow going away?
No, not at all. I mean, it's typical for most respiratory viruses that your antibodies wane over time because you don't have to be. It's been seasonal for most of us living not in a single season environment. So I think it's very typical to have this fluctuation. The memory cells are created. There are different types of memory cells. There are circulating memory cells. There are tissue resident memory cells, which are specific to the structures where this immunity is needed the most, or they can go back to the corresponding lymph nodes a little bit deeper, into the tonsils, for example. And there are long-lived immune cells which home to the bone marrow. And in some cases, people can take samples from the bone marrow, and 8 months later, these, despite having no measurable antibody levels in the blood, these B cells, these memory B cells in the bone marrow, could be shown. So it doesn't mean that you don't have immunity. And then we also have memory T cells. And in the respiratory system, it was shown repeatedly that the persistence of memory T cells in the specific tissues requires longer-term exposure to a pathogen. So a single mild infection may not result in long-lived memory T cells, but if your body recognizes the virus as a really novel pathogen, which is described in several people in connection with SARS-2, then the body retains some of the viruses or some of the viral proteins for this kind of immune maturation. The body retains some viruses or retains some viral proteins and continuously feeds these pre-memory cells with these antigens so that they mature into longer-lived tissue-resident memory T cells. And this was shown to occur in connection with SARS-2. And from previous research mainly on influenza, we know that long-term antigen exposure results in longer-term memory T cell generation. Or most likely, repeated exposure can achieve the same. So if these are seasonal cold viruses, like coronaviruses, for example, then you are exposed almost every year, and then these memory T cells persist in your respiratory system. So what happens during a kind of a reinfection, or let's call it re-exposure, because you may not experience any kind of symptoms for the second time, that the virus again arrives, and even if it successfully infects a few cells, the memory cells are there, and what took 1 week to clear for the first time or 2 weeks, it happens in 1 day or 2 days. So it's a very big difference, 1 week or 2 week disease course, or 1 day or 2 days. And of course, if it's 1 day or 2 days and you have no symptoms, you are much, much less likely to spread the virus. So upon reinfection or re-exposure, I prefer re-exposure because infection requires the proliferation of virus to levels that you could transmit or spread the virus again. So upon re-exposure, what happens is that you clear the few viruses in a day or two, and there is no transmission at all involved.
This could be a good point to take up the whole idea of asymptomatic transmission, because I mean it would be interesting to get your views, since you were saying that if you're reinfected the second time and you don't get symptoms, then it's not possible that you would spread, or it's highly unlikely, to say the least. So
I think that transmission without symptoms does exist. So it's not a myth completely, but I think even Fauci admitted 1.5 years ago that asymptomatic transmission is not the driver of epidemics. So it does occur, it contributes to transmission, but it's not a driver. The main driver typically is when people, for example, sing together or party together or go for a smaller shop where the ventilation is not so good. And spend some time together, even just talking or singing or whatever, and one of them or a few of them release huge amounts of these fine aerosols, and the others inhale and get infected. This is the most typical. That some people release a lot more of these droplets than others, and those kind of super spreaders infect dozens of people, while the other people, 10 other people, infect one or nobody. So this is the typical distribution. And I think that most asymptomatic transmission occurs before symptom onset. So it's in fact a pre-symptomatic. So these viruses can suppress immune responses. An immune response is responsible, for example, for the pain or the mucus, the excess mucus production associated with symptoms. So when you have this sore throat or you develop a cough or just some other heavy mucus production, this can be missing during the initial part for 1 or 2 days. And then this 1 or 2 days, when the viral load is already high enough for transmission to be incorporated in these fine aerosols, but you don't really feel the symptoms yet. This is where pre-symptomatic transmission occurs. I think that's and these viruses really evolve over time to cause milder and milder symptoms, and to extend this time when you don't stay home and away from other people, because then they cannot spread. So it's a natural way of evolution, that natural route what they take, that milder symptoms, longer pre-symptomatic transmission. It's not miracle. Some people deny that this exists, but this is what the viruses really have to evolve to do, because otherwise, if you are really sick, you have after only 1 day, you have a really sore throat, and your head is full of the excess mucus. You cannot see, you cannot breathe. You just stay home and don't go among other people, and then you cannot spread. So this is not what promotes transmission. And if it doesn't promote transmission, those viruses which do this will just die out, or just will be much lower in frequency. So I think this naturally occurs, and in fact this is a route what most respiratory viruses take over the long term, that they evolve to be less symptomatic and a bit longer pre-symptomatic transmission. I think this is also what has been happening to SARS-2. The so-called variants of concern are all creating viral loads initially, and then before symptom onset you can spread to more people. This is what they have to do in order to survive.
Yeah, from my own review of the scientific literature, I have read quite a lot of studies on asymptomatic as well as pre-symptomatic transmission. But pre-symptomatic transmission is not a term that's commonly used, and pre-symptomatic and asymptomatic are lumped in together. But from my understanding of this, I think it's very, very crucial to distinguish between both, because the PCR could pick up dead viral fragments in someone probably a long time after they've had exposure. Like we've seen this for measles and MERS and things like that. That people could test positive for the viral fragments that are still left in the body long after the infection is passed. So from the point of assessing if someone's movement should be restricted, I mean, like from my perspective, like even if we could prove that someone has active, say, virus in them, I still don't think it makes a case for restricting someone's movement or taking someone's liberties away, because of the kind of data we have now about the severity of this and all the things we can do with respect to treatments and prevention, and the different the age differential, and the differential with respect to comorbidities. I don't think that restrictions on movement are justified, even if you do happen to find some virus. But why don't you kind of go into the PCR now, and maybe talk about like how it can't really differentiate between this asymptomatic and pre-symptomatic thing you're talking about?
Yeah, I think first we should touch upon this kind of restriction of movements.
Yeah.
So this comes back to the very early recognition that the disease is very different in elderly, frail people and in young and healthy people. So why to restrict the movement of young and healthy people as long as they restrict their movements to stay among themselves? So let's arrange something which could somehow reduce the mixing of young people with the elderly people. Of course, I do understand that it's not possible everywhere, and the local situation is different, because in some places, for example, in Mumbai, a lot of people live together in very small closed spaces, like millions of people in a part of the city. So it's simply impossible to restrict their movements or separate them, for example. But in other places where very old and frail people live, mostly in care homes, for example, in Western Europe or in the US, this could have been done. That restricting the movement of the elderly and visiting and carrying the elderly around in hospitals. So some hospitals or hospital sections should have been separated from other parts of the hospitals to completely prevent the mixing of old people with other ill people. But this wasn't done. Just the opposite. Very often in the UK, for example, or in other countries, in the US, the government sent the elderly people from hospitals back to the care home, and they carried the virus back and then spread it all around. So that was the stupidest, the most stupid thing to do. Okay, so the PCR, yeah.
Also, just one thing I'd like to add on that. I think this whole mindset of restricting movement and trying to stop transmission also stems from the fact that there's a vaccine on the horizon. So we need to do that until the vaccine's out, because the vaccine is safer than getting a full-blown infection. But I think that kind of thinking doesn't really take into account the pitfalls of the vaccines and the adverse events also that could arise from vaccination. So,
yeah, vaccines are clearly a preventive measure. So it's not what we typically start administering during a high spread season, like late winter, what happened early this year. And there is a reason why vaccines are not used during the infectious season. For example, for influenza in the Northern Hemisphere, the season starts in late November or something like that, and until early March. And vaccination starts in September. So it's September, October is a typical vaccination for influenza before the season. delivering vaccines during the season, I think it's counterproductive, especially if these vaccines are very strongly immunogenic, because then they cause a certain level of immunosuppression, and then your immune system becomes even less competent to deal with the infection than before vaccination. So following vaccination for a couple of days, your immune function decreases. So your chance of getting or developing a severe disease is considerably higher. In population studies, it was shown, for example, in Denmark, that in healthcare workers, their chance of developing symptomatic disease was 118% higher, more than doubled, following the first week of after vaccination. So it's not a good idea for this reason at all. Otherwise, these people should be completely separated. But what if they had been previously exposed to the virus and the virus is still in them in a kind of a suppressed state, and then you suppress the immune system which controls the virus, and then it just suddenly explodes. So this is also how to create super spreaders. Because super spreaders are usually immunosuppressed people to some extent. If your immune system is not very competent, then you develop higher viral loads, and the higher viral loads makes you much more suitable for delivering high puffs of aerosols full of viruses. So if you suppress the immune system with a vaccination, a strong vaccine, a strongly acting vaccine, then that those people are exposed to the virus, they can become super spreaders very easily. We have plenty of anecdotes. I don't know if there is a single study looking into this, how the family of vaccinated people become a complete disaster of COVID just in a week following vaccination. So the elderly family members got the vaccine back in January, February, and then all their family developed COVID within 2 weeks.
Also, this point is interesting to highlight here, specifically with the PCR that you'll be speaking about, that the symptoms of SARS-CoV-2, of course, overlap with many kinds of viral respiratory infections. So even when we are testing, at any point in time, you're not really ruling out for other things, right? Like if you're just doing a PCR for SARS-CoV-2, you're not really looking at the many infectious viruses that could be present. So is that really a prudent of infectious? Yeah.
Technically, PCR is not suitable for detecting infection. So infectious people. Not even a person who is in a active viral phase of an infection, because all what it does is finds and multiplies viral RNA, viral nucleic acids. So, but we could do something to improve the correlation between a viral load, which corresponds to potentially being infectious, and the PCR testing. So it was not so difficult to find out already spring last year and point out that the cycle threshold, which is how many cycles you need to, how many times you need to double the amount of viral nucleic acids so that it gives a positive result in the test. And of course, it's not so difficult to point out that the higher the viral content in the original sample, the lower number of doubling is required for getting a positive result. But this is not perfect, the cycle threshold in itself, because it depends on how the sample is taken. If some nurses who take the sample are more precise and they scrape out more viruses from the same person. If another person takes the test, you can see much lower signal, for example. Much less viruses were were cleared with the swab. So it's there are different factors contributing to this, and it's not quantitative for this reason, but very low cycle thresholds always mean high viral load. That's not controversial, and very high cycle thresholds can be whatever, very early infection, can be a bad sampling, poor sampling practice, or can be the lag phase when you don't have live viruses anymore, but you still clear out some cells after infection. So, but then just do a second one. So leave a gap 24, 48 hours and do a second PCR. And most likely you will get a second person taking the sample. You will get a second result which can show higher viral load, then you could be infectious, so stay at home. Or you see the same very low level of viral load, then you are in the lag phase, because if 48 hours later you still have very low virus load in your nose, then you are not infectious, most certainly. Or it can completely disappear because you are even out of the lag phase 48 hours later. So then if in doubt, do a second test. Or use multiple quick tests, the lateral flow assays, because lateral flow assays look for viral proteins. So viral proteins are only released as long as you have active infection. 48 hours later, you don't have viral proteins in your, and the sensitivity just happens to be the level which you need for the productive infection, and it correlates very well with infectiousness.
Yeah, I think I've seen many papers myself where they compare antigen tests as well as the PCR to viral culture, and they segregate according to the cycle threshold as well. And in many papers we've seen that that the likelihood of culturing highly reduces, like culturing an active live infectious virus, which is what is actually needed to cause an infection in someone, that strongly reduces as we start to go above 25 cycles. I mean, this has kind of been shown in meta-analysis as well as in Jafar's paper as well, that I think Portugal's courts had cited when they kind of struck down the PCR. So why, just with your views on pre-symptomatic and asymptomatic transmission not really being the driver of pandemics, and that being, I think at least it's fair to say that it's very rare, if not non-existent, that in terms of actually driving the spread of disease. So my question to you is, like, instead of using these tests that have a high rate of false positives compared to viral culture, both the PCR as well as the antigen tests as well, why don't governments start using the viral culture test itself, which these they're comparing it to in these studies, and start using that on symptomatic people, to kind of restrict movement?
Yeah, I also don't think that viral culture should be used for detecting infections. On a mass scale, because viral culture is really cumbersome. It's slow, so you don't get back the results for many days, and it cannot really be done in the scale which was done on. I think, well, it basically boils down to whether there is a need at all to try to follow the spread of the virus in a mass scale. Actually, I don't think so. That there is. We can do what we can do at a relatively low damage to life, damage to economy, damage to other healthcare problems, other healthcare services, with rather straightforward measures. So if we really need to slow the spread universally, which I actually don't think is a good idea at all. If we really need to slow the spread, just ban indoor gatherings of people, especially in poorly ventilated spaces. So stop the movies, the theaters, the whatever. and remove the old people from these places. So ban the old people from the shopping centers, although modern shopping centers, modern cinemas and theaters have quite good ventilation systems. So it could be set up with a simple UVC light to disinfect or remove any nucleic acid containing pathogens from the air. So the air could be treated, it could be increased, the ventilation could be increased, and then the chance of infection, even in these places, is low. But
Yeah,
then
but you're of the opinion that that shouldn't be done and that it's not really prudent to try to stop transmission. Could you elaborate on why you think so?
no, I mean, we got, there was this great Barrington Declaration.
Yeah.
And what they said was that we should have separated or isolated the most vulnerable people, and then let all the others live their lives and build immunity.
Yeah.
It's not really. Others interpret this as let it rip. This is not the case actually, but eventually, let it rip happened with or without measures. So those people who say that this is irresponsible refer to a idealistic, non-existent scenario where we completely eliminated viral spread and nobody died and we everybody is vaccinated and so on. And then they bring up crazy examples like New Zealand and so on. That this is not applicable anymore, because if you had the strong early spread like in Western Europe or North America, it's impossible, because now we understand that the virus is in cats and dogs and deers and minks and all kinds of animals. So it was not possible from the very beginning to completely eliminate the virus in Europe or in the US or in Central Asia or in many, and especially in developing countries, they cannot afford, and the threat was also much lower to them. And the other argument was that you cannot isolate all elderly people. Yeah, if you send them back and forth between COVID wards in hospitals and care homes, that's the worst possible thing to do. But that's not what was proposed. Interestingly, my idea was to accelerate spread among the young and healthy, because the quicker you can immunize them, the shortest time is required for isolation for the vulnerable.
Yeah.
So that's the least harm that could have been done. So accelerate. That's why I was thinking about these camps, kind of vacation camps for kids and young people. I mean, all concerts have been banned, and these performers suffered great financial loss, and so on and so forth. We could have paid the concerts for the young people. We could have even paid the first drink or a couple of drinks for these people. And this would have been still a lot, way cheaper than doing this crazy thing of stopping the world for weeks, and then it didn't solve the problem. Let's stop us again the world for a couple of weeks more, and then again and then again. It's just completely crazy. I mean, just let the young build immunity. I volunteered my kids, so I said that I'm not crazy. I'm talking about this in a way that I'm ready to send my kids to these camps. I have teenage girls, and they are very, very, very low risk. They are healthy, normal weight. They are vitamin D replete and nutritionally replete. We could have even done some correction nutritionally or vitamin D from a vitamin D perspective before starting this, but it should have been started as early as possible and as quickly and as targeted, and in a controlled way. Because doing it in a controlled way, I mean, limiting the viral load with a spray, a small load, and then let mild disease develop or something like that. They could have even stayed asymptomatic or whatever, and do it in a controlled way with medical supervision is much better than just leaving it and let it spread. It's just the opposite. It's not let it rip. Do it in a controlled manner. Accelerate among those who are not vulnerable, and slow among those who are susceptible to severe disease. That
I
was my
think it, yeah, I mean, I totally agree with you. And I think it comes down to like individual choices, right? Like if I'm okay, if I think my risk is fine, if I'm going to go and contract this and I'm okay contracting it, like, why should the government stop me from doing that? If I want to get infected fast and then develop
yeah.
natural
But
immunity, like that should be my
this should
choice,
have
right?
this even should have been incentivized, like what they do with the vaccines now. They have lottery,
Yeah.
vaccine lottery, and whatever. They are handing out money and hamburgers and whatever you can imagine just to vaccinate yourself, despite previous immunity by infection or whatever. So, or extremely low risk for kids or adolescents, for example. So they are doing it 15 months late. It should have been
Yeah.
done for immunization for the young people. I mean, exclude obese, exclude diabetic, and those known for immune problems and whatever, and all the others provide incentives. Arrange them these big concerts in closed places with turned down ventilation, and then they can have fun and at a very low risk. And the risk
I think,
is the same if you get it in your school class or get it in a supermarket or whatever. The risk is absolutely the same, or even better because it's done under medical supervision.
yeah, I totally agree with you. I think this whole mindset of trying to stop the transmission is based on, I mean, at least was based on the idea that the vaccines are going to come in a year and a half. So we need to wait and hide till then until the vaccines are out, and then those could be given to everyone. But of course, I mean, we would stand against mandates of vaccines of any kind. So if that anyway is going to be a choice, then like shouldn't the people be given a choice to contract the infection and go that route? And even, I mean, talking about the obese or people with comorbidities, we have, like, I mean, really good science now on different kind of medical interventions or even preventions with respect to vitamin D and things like selenium or melatonin, and with respect to treatments, so hydroxychloroquine, ivermectin, the math protocol. Like, we have so many things that we could do even if someone does get infected, if we act early enough. So I just think that this whole like stopping the world, it didn't make sense because it was based on the idea that we need to keep people in hiding until the vaccine comes out. But if you can't mandate the vaccine, then if we would stand against vaccine mandates, then just let people make their own choices. Like, you can't stop a respiratory virus from spreading once it's out. Like, it's just going to go and do its thing, right?
Yeah, it looked like that no economic, financial, and mental and other health loss was a problem just to wait until the vaccines come. So it's not a problem if you die of another disease which could have been prevented by not hiding at home, for example.
Yeah.
That's perfectly normal, but developing a mild to moderate disease from COVID was absolutely unacceptable. So if I had a choice of developing a mild course of COVID versus hiding at home for a year and potentially getting a heart attack if I'm prone to that, for example, or if I miss a cancer diagnosis by a couple of months, that's a very different thing to happen, or postpone a hip replacement or whatever procedure by a year or even more because of this. Yeah,
Yeah.
it's. I think the world just went crazy.
The adverse effects of the interventions weren't really taken into account, and there's this undue focus on just COVID deaths, and I mean, kind of ignoring everything else. So I just want to ask you the question, like, what do you think are the odds? Because we've never done mass testing in the world, right? Like, let's be honest, we've never done this for any other kind of infectious agent or pathogen. So, I mean, this whole thing started out with identifying people who apparently had a different kind of pneumonia in Wuhan, and then finding a novel virus out there, and then doing that all over the world. So, what do you think are the chances that a lot of people who actually had respiratory infections but had them from other viruses, since we went and mass tested them with the PCR for this thing, we started finding this when probably, I think it's fair to say, in many cases, the SARS-CoV-2 was not actually behind the symptoms, and it probably was other kinds of respiratory viruses. Because we know, I mean, I don't know if you've read this or not, but we know like a similar case happened in Dartmouth Hospital in 2006, where they suspected that there was a whooping cough epidemic, and they went and quickly tested everyone with the PCR and gave everyone whooping cough pertussis vaccines. But then later, there were some scientists who actually sent the samples to the CDC, and they figured out that there was no culturable whooping cough, and it was actually something else, and they created a fake epidemic. So, do you think it's a possibility that this thing has happened all over the world, and a lot of different respiratory symptoms have been dumped into this category because of that positive PCR test?
I don't know. I'm sure that there was this virus was spreading much earlier than what we believed. So perhaps even the October 2019 is a very, very pessimistic scenario, or it may not even have been the first such virus spreading. There are some speculations based on the characteristics of the virus that this is a descendant of a vaccine attempt itself. So it's a spreading vaccine, at least, or if not this one, then one of its ancestors in a lab. And what if the ancestor was released first as a vaccine attempt, and this one only escaped or released later? So there are a lot of speculations, but those are speculations. What the data shows is that the virus was spreading already in December in Western Europe, in North America, and in other places. So by the time we recognized it, it was late February or early March.
But we do know that there are other, viruses that you could pick up. Like, I've read many studies in healthy people where they test healthy people's viromes, and they find all different kinds of pathogenic, say, viruses and bacteria, and they're not necessarily contributing to infection. So just doing this, what I'm trying to get at is just testing one infectious agent's fragment with a PCR and saying that the person is falling sick from that, when we have since I look at infectious panels for patients also, like when we're doing gut testing or something, you have like a large array of microbes and the PCR targets when you're trying to evaluate, say, parasitology or the bacterial infections in someone's gut. Like, typically you have multiple things testing positive together, right? So like what should have been the approach for diagnostics? Like, should we have included other respiratory targets along with the PCR? Would that have been more appropriate in terms of diagnosing respiratory infections in people?
Normally you test people when they have symptoms.
Yeah.
And they have typical symptoms, but respiratory symptoms overlap a lot. So many symptoms are not specific to one virus or the other. So it's a difficult question, but I think the test itself, at least the later iterations, the first few, may not have been the best what we had. But later iterations are specific enough, so they pick up a certain virus, and if it's accompanied by the typical symptoms, it's most likely a infection by that virus. Also, if there are really epidemics, these respiratory viruses tend to suppress the spread of other viruses. It was also shown that if you had a cold just before a wave of SARS-2 in that region, for example, you had a rhinovirus, a typical cold virus infection, you were protected to 90-something% from symptomatic COVID, because your immune system is already alerted in the respiratory system, so in the airways, and the virus has no chance of quickly proliferating to symptomatic or transmissive levels. So prior infection can indeed suppress other viruses. That's also known from previous research, that these viruses come in waves, and even their season is different. So one type of respiratory virus arrives already in November in the northern hemisphere in temperate climates. then the other follows in January, and the third one, typically coronaviruses, start only in March, for example. So this has been known previously, that they only partly overlap, because they use the same susceptible hosts for infections, and if one makes the host alert to respiratory infections, then the next one has no chance.
Yeah, I mean, of course, that's a very plausible, sort of view that you put out. But I think we wouldn't know, right, unless we test for the targets. Like, we wouldn't really know what's responsible for someone's infection. And I get it makes sense that if it's a novel pathogen and if it takes over, then other viruses would kind of be, not a co-infecting at the same time. But I think that, I mean, mechanistically, of course, it's interesting, but it would be nice to see like real world data about, I mean, like even the CDC now with their last advisory, they're kind of probably developing an assay to differentiate between influenza and SARS-CoV-2 at the same time.
That is gonna be another terrible terrible idea. I mean, if you are actively looking for something, you most definitely find it.
Exactly.
This happened. A good example, what happened in 2003 in British Columbia, I think, in Canada. That was the era of SARS, the first epidemic, and right after, during the summer, there was an outbreak with SARS-like symptoms in a care home in Canada, and the big healthcare team arrived with testing, PCR, everything, and started looking for SARS, and they couldn't follow. Then they launched a multiple test which looks for different viruses, and then they found that the outbreak was caused by a common cold, the so-called OC43 coronavirus, and it just went through the care home and killed, I don't know, something like 8-point-something% of the infected and symptomatic elderly. So you can have a high infection fatality ratio in a care home where a lot of vulnerable people are condensed, but from a simple cold virus, because they are susceptible to such diseases, and it can be detrimental even if they are infected with a so-called common cold virus.
Got it.
And if you look into it, otherwise, what could have happened if they don't go after the virus this way with the PCR? They just account the outbreak that there was some pathogen and there was some pneumonia developed, and this number of elderly unfortunately died of this pneumonia. And then the bookkeeping goes to influenza or pneumonia, and that's it. And then there is no PCR and there is no access test. Basically, that was just regular deaths, because this happens from time to time in care homes or in hospitals or in places where these people are happen to occur in concentrated numbers.
Yeah, and that kind of adds to my concern that a lot of the people who've been lumped in the COVID category could be, just from a regular cold virus or something, and we wouldn't know until we test. So that does make sense. Yeah. Finally, just, I mean, to wrap up this, at least this element of the discussion with the vaccines and the testing. Two things, if you could just elaborate on the superiority of natural immunity over vaccine-derived immunity and the science around that, and probably address, because there have been some people who've claimed that the PCR is not specific enough and it can't differentiate between influenza and coronaviruses and SARS-CoV-2 and variants. So do you have any insights on the second question, and then maybe you could go into the natural immunity, vaccine immunity side of things?
So what would you like to know about the specificity of the PCR?
Yeah, some people have claimed that the PCR is not really specific and it can't distinguish between influenza and coronaviruses and things like that. At least according to my research, that's not true. And I know that if the primers are designed well, then you could differentiate. Yeah.
I'm not there personally checking everything, so I cannot be 100% sure. But based on my understanding, there were some issues with the first tests, especially pushed through without proper review of the studies and so on and
You're
so
talking about the
forth.
common Drosten review, right?
Yeah. For example, also some of the CDC tests approved or developed tests were not perfect. But and also initially there were some problems, constraints on capacity, and then looking for 3 fragments is the gold standard, and then they gave up and looked only for 2, for example, or some I heard even only for 1, which is basically rubbish. But after ramping up capacity and fixing these initial problems, I think that the test is really specific enough for the SARS-2 virus, and there is very little overlap and misreporting of so-called cases. I mean, positive results. And the bigger problem is interpretation and use of the results for creating guidance for pandemic management. That's a different story.
That's interesting, because I've reached a similar conclusion also, because I myself wrote a paper on the PCR and the faulty interpretation of that. I think, of course, the early issues were there with the assay design and all, and I think Bobby Malhotra and Kevin McKernan and some people wrote an entire paper on that. But I think later on that was kind of fixed, and the gene targets that they were looking at were very specific for SARS-CoV-2. The problem really comes in is the significance of the PCR. Like, what does it really mean by just testing positive? Because we know that it could very well be picking up an infection that someone previously had, and they're not a threat to anyone. So, what's the point of counting them as a case and restricting their movement? So I'm fully with you on that, and it's nice that like your views align with mine. So yeah, I mean the second half of that was probably go into the science of natural immunity and how that's superior to like vaccine derived immunity, and probably we could even share my screen and go through some of the studies that you've highlighted, which kind of talk about the importance of natural immunity.
Sure. I mean, the very basic difference between naturally acquired and vaccine or gene therapy induced immunity is the route of administration. So normally what you experience is, we discussed this earlier, that you inhale the fine aerosols, and then the viral particles reach your lung or respiratory airway cells, infect them, trigger a signaling cascade, and then the signaling cascade recruits the immune cells, and when the inflammation and the viral phase is resolved, and there is some repair phase going on, and then it's a complete resolution, and you have the memory cells, which are the kind of the bastions of the next response in the airway mainly. And then you also have some some memory cells circulating, and some memory cells even home to the bone marrow. And compared to this, when someone is injected with, for example, a mRNA or a viral vector, so-called vaccine, then it goes into your shoulder, into the muscle, and it causes some inflammation, local inflammation in the muscle, and there are some immune cells recruited, of course, to the place of inflammation, and they take the antigens, and probably some of them are also infected and take some. So some of those will produce spike proteins in the lymph nodes, and then this antigen is used for creating antibodies, and some T cells, more CD4 plus, so more effector or regulatory cells than the CD8 plus, so more less killer type of cells, because they don't meet so many viral proteins which trigger these killer cells. I think that is, for example, one of the biggest trigger for these killer type of T cells is the nucleocapsid protein of the virus, which is not injected or not generated following the injection. So this CD8 plus arm of the T cell adaptive response is much weaker than the antibody producing cells are more systemic, because you were injected and not inhaling the antigen. So you need really high levels of antibodies, IgG, for example, so that it gets secreted into the airways. It does get secreted into the airways when the systemic levels are high, but when these levels go down, you stop secreting these IgGs, and then you have no protection in the airways. So the very early and very specific protection you tend to quickly lose, and that's why the enzyme, the vaccines are not really, these types of vaccines are not really a good tool to develop sterilizing immunity on the longer run. So you may have sterilizing immunity for a couple of weeks or a very few months, like 2 months or like 3 months perhaps. But after that, your antibody levels systemically will drop to a level which will prevent the release of these antibodies into the airways, into the mucus. So then you are not protected in a way that you cannot transmit the virus. So it depends on who you are measuring. If you are measuring, if you are doing these efficacy analysis shortly after vaccination, they show much higher efficacy than 3, 6, 9 months later. That's why everybody is talking about boosters already, because some people received the second dose already 6 months ago, and their levels, especially that these were older people who or obese people or sick people, they tend to develop weaker immunity by default. This was shown in influenza, for example, that obese people mount a much weaker immune response and lower antibodies and more quickly waning antibodies. This is, I'm certain, the case in connection with SARS-2 as well. So, yeah, the boosters may have a real role to play, but why don't we develop proper vaccines instead? I mean, we could develop, and there are some candidates, but sooner than later, nobody will remain to test on. I mean, by this time, we see 90-something% antibodies, for example, in the UK. When then you have representative tests for antibodies, now it's 86, 90-something%. So there is nobody. And then even the remaining few percentages could have been exposed, but they don't have the high levels of antibodies which hit the threshold. So basically, almost everybody has been exposed. So there are no people left to test the vaccines on. the new vaccines. There could be much better vaccines by design, and it was known. But the most important thing was that develop something the quickest possible and not the best possible, and the most patentable, so that you can squeeze out the last drop of money from the governments was the important thing, and not the highest quality vaccine design. I think that we could handpick a dozen or so proteins, protein parts, polypeptides, protein segments from the virus, including those not translated after vaccination, because when you have an infection, not everything goes perfect. So when the virus proteins are made, sometimes the process doesn't start at the beginning, or there is just a shift. You know that 3 nucleic acids code for an amino acid, and if you start not reading by the first, but the second or the third, there is a total shift of amino acids get generated. So there is a protein or protein segment generated, synthesized, which has nothing to do with the viral protein. Actually, it's a completely novel protein segment, which is still produced during natural infection, and our immune system is responsive. And some of the most immunogenic proteins are these so-called mistranslated or out-of-frame translation proteins, which are very specific to natural infection. And you can find this in my stream or on my wall on Twitter. If you search for out-off-frame, you will find those studies, that small thread that shows that these proteins are specific to natural infection and are very highly immunogenic. So, and these are not autoimmunogenic, for example, because these are not present on the virus. It's not part of the virus normally. So when you have the virus going around, or a spike protein, for example, going around, and they have certain level of similarity to our own proteins, you can develop these autoimmune responses. But these out-of-frame translations are not autoimmunogenic at all, because they are not coming from an adaptation of the virus. These are errors, basically translation errors. And we could handpick a dozen or so of these protein segments. There is a receptor binding domain, the N-terminal domain, some part which is not autoimmunogenic, the parts of the nucleocapsid, parts of other NSP3 or 13, whichever was shown to be very highly immunogenic. Those parts which are not autoimmunogenic but highly immunogenic. And then this polypeptide mix could be used for creating nasal sprays, and just use as sprays into your nose. Use the natural way of infection for immunization. I think those would be superior, and those could be administered repeatedly without any problem. So you could get it even during the same season. If there is another wave, so-called wave, then you just see that it's starting again. You just do the spray again, and you are protected, and others are also protected. And there is no chance of problems, systemic problems, no myocarditis, or no dissemination of the lipid nanoparticles, or dissemination of the spike, highly inflammatory spike protein, and this kind of problems could be totally eliminated.
Got it. That makes sense. I've also looked into James Lyons-Weiler's work, and he was kind of suggesting the same thing as you. He had written to the CDC, I think, last year, talking about how they shouldn't really use the spike part as the target antigen, but probably look at other targets that you were talking about, mainly that don't elicit these kind of responses. So of course that does make a lot of sense to me. Also, do you think there's any evidence that like just talking about the current vaccines, like we know it's just targeting the spike, so how would say immunity to that compare to naturally derived immunity? And we know there are some papers on that, so maybe you could talk about that.
Yeah, I think there are more or less three issues with the current vaccines. One is it's spike only, and then this problem can already split up to different sub problems. Because the spike itself is unusually inflammatory. It contains receptor binding sites to, for example, TLR4, which is a more typically bacterial sensor.
That's the toll-like receptor, right?
Yes, yeah. The TLR4. It's on the S1, as far as I know. Then the spike also contains a bacterial superantigen, which is an extension of the furin cleavage site, and which seems to work as a bacterial superantigen. This may be one of the pathways for developing the weird symptoms in a few kids who develop this long-term MIS-C or whatever it's called. And the spike contains a heparan sulfate binding site, so it's partly responsible for the coagulation problems. It was also shown to aggregate lipopolysaccharides. That also signals through the TLR4. So it's kind of the convergence upon a single, usually bacterial, inflammatory signaling pathway. So it's very highly inflammatory, the spike in itself. Then, of course, the breadth of the immune response is not similar to what we see following natural infections, because several viral proteins, at least 3 other viral proteins, were shown to be highly immunogenic. The others are not so much. And then we have these out of frame translation polypeptides, and that's a problem of the spike circulating around. And then we have the problem of the lipid nanoparticles, or the adenoviral, the virus vectors. The viral vectors implicated in the thrombosis, the coagulation problems, especially in younger women. And the lipid nanoparticles are theoretically, the positively charged ones are also TLR4 agonists, so they cause inflammation through the same pathway. I'm not sure this is really a problem in vaccination. Perhaps it just contributes to the inflammatory response, so it does the job, basically, the job of the adjuvant in the mRNA vaccines. And what else?
Did you think there's any additional benefit that could be conferred? Like, if I have natural immunity already, do you think that it makes a case for,
Yeah, we already discussed the kicking out of the innate immune system in specifically in the respiratory, in the airways. When you meet a virus, it's a different response when you are injected in your shoulder.
Yeah.
The not generating memory T cells, for example, in the airways specifically, and especially not the killer types of memory T cells. I think these are obvious drawbacks of current vaccination strategies.
Yeah, so I think, just keeping all of this in mind, it's really disingenuous that natural immunity has been ignored and all the focus has just been given to vaccination, when there's so much robust science as well as so many papers on exactly how natural immunity works, and the depth of the efficacy that it describes.
We know a lot about what happens after respiratory viral infection. So it's anybody who believes that it's fundamentally different how the immune response works following SARS-2 exposure compared to influenza A virus exposure. There are certain differences, but it's not fundamental. It's fundamentally the same.
So do you think there's any justification to the idea, like governments are telling us, even if you've got natural infection, like just go get vaccinated again, because that will boost your antibodies and give you better protection? Do you think there's any science to that?
I think it's completely bogus. So there is no scientific reason, if you had a mild infection, to get vaccinated. There is absolutely not. People tend to believe, and even some so-called scientists fall for this, that if you get a vaccine following infection, then your antibodies go up.
Yeah.
And I say, so what? Antibodies in my arms, because they take the blood from my vein in my arm or in my legs. Why do I need higher antibodies in my legs or arms or stomach? Please explain, because I just cannot follow. I need high antibodies in my secretions, in my saliva, in my mucus, specific antibodies against SARS-2. So the next time I meet the virus, then they bind it and lock it into the mucus. So they can prevent infection and provide even sterilizing immunity. I couldn't care less about my antibodies in my blood. So because for the second time, we just don't expect systemic dissemination of the virus. If you have been exposed, you may develop mild disease at worst if you have still a functioning immune system. No systemic dissemination of the virus is expected. Absolutely not. So why systemic antibodies is the focus is totally scientifically bogus. We should be looking for memory cells in the respiratory system. We should be looking for immunoglobulin A in saliva and in the mucus and these kind of things to establish the state of immune memory after infection. That's how the system works, and that's how we need to look for these markers. It's not immunoglobulin G circulating in your arms and legs and wherever. It doesn't really matter. Even if you need them, I mean, if you had a moderate or even severe disease, but you survived, then you are probably more prone to developing a symptomatic disease the second time, because you are immunosuppressed, you are immunocompromised to a certain level. That's why you have strong symptoms, a strong reaction for a respiratory virus. So next time, or you may even develop some other disease over time. You got COVID spring last year, and since then you gained 10 kilos, 25 pounds of weight, and you are almost diabetic now, and you are more prone to developing severe disease. So you will have a symptomatic moderate course of disease, and you need systemic antibodies. Okay, but what happens then? You have your memory cells in your bone marrow. They start proliferating and release plasma blasts, and that these will produce the necessary antibodies what you require. So it's just 1 day of delay. It's not like the last time when you struggled for 2 weeks. It's just 1 day of delay. So you are going to develop a mild disease typically for very few days.
Great, Gabor, man. Thanks so much for sharing your insights. I know we went too much into the weeds out here this discussion, but this is something that I specifically wanted to do for viewers, because I've given the viewers a broad overview that, most of the positions we discussed that I already hold, but I got Gabor on to go deeper into the science and to probably, show the viewers that there is strong scientific literature, and we have a lot of knowledge on natural immunity as well as testing that, the mainstream media or the governments don't necessarily talk about. So thanks so much for sharing all those insights with us. And thank you so much for being so kind with the time also. It's been almost 2 hours, and I was planning to go into the other diet and lifestyle kind of things, but probably I could have you on a second time and we could just do that. But just let viewers know where they could find your work, probably find some of the things you write on a regular basis if you do, and also just give us a wrap up on, your whole position on this pandemic that's going on and what you think should happen.
Yeah, just a moment, and I change my name on screen for my Twitter handle, and then you can easily find me. The wrap up is that I think I have had the feeling for almost 1.5 years that science has been hijacked, and the communication from authorities and the World Health Organization and other organizations serve not the interest of the people, but the interest of certain groups and corporations. I don't know why and what the ultimate goal of this, if this is really about fighting climate change in the long run or whatever. I don't know. There are a lot of theories out there. I'm not really into the politics or the long-term societal problems. What this causes or what the background is. But what I'm really concerned about, how easy it was to hijack the scientific community and the scientific communication behind this pandemic. It's totally shocking, in fact. So that's my very brief conclusion.
If you could just briefly summarize your positions on the lockdowns, on masks, on testing, on vaccines. I know it's difficult to do, but if you could just like give a
Yeah,
brief.
lockdowns is not really my expertise. I read a few papers from before the pandemics, and this has never been a viable option. And as we see, I think that the outcomes also prove that this was totally bogus science, and it caused a lot more harm than what it may, but what benefit it may have delivered. So from lockdown wise, from masks and sterilizing your hands and these kind of things, yeah, I was my first days in the OR already informed me that this is not a realistic preventive measure. For certain people, really vulnerable, they may try with very high quality masks or respirators to protect themselves, but without even putting a quality or guidance on how to wear, how to replace, what types of masks are worth wearing, if at all. This is only virtue signaling. This is not science. I mean, there are huge differences between some cloth masks and even some cheap surgical or medical masks compared to very high top quality surgical masks or some respirators. There are extreme differences between filtering ability, and even the best respirator is totally useless if you don't know how to wear it, how to replace it, how often to replace it, and these kind of things. Without this guidance, this whole masquerading is just virtue signaling. It's theater.
And
Not,
that whole narrative totally ignores the downsides and the side effects of prolonged mask use, which I've read quite a lot of papers on, that there are real legitimate risks to prolonged mask use, and certainly they might have a time and place in certain contexts, but overall mandates are totally unethical because anything that has a risk should have choice.
yeah, but this has never been about your health.
Yeah.
So
All right, man. Thanks for letting me pick your brain. It was a
yeah,
pleasure
thanks for
speaking to you.
inviting.
Yeah, I think if I would encourage viewers, if they want to kind of go and explore the studies or the science we spoke about, then Gabor does tweet extensively and writes his views on a lot of those things. So I encourage go follow him on Twitter and his handle that's shown on the screen. But again, yeah, thank you so much, Gabor, for coming on. Pleasure to have you.
Thanks.
Hopefully, we can have another conversation about the other things that we spoke about. Thanks.
Okay. Bye-bye.
Bye.